बिस्मिल्लाहिर्रहमानिर्रहीम अस्सलाम वालेकुम स्टूडेंट्स। आज हम डिस्कस करेंगे अबाउट द वेरिएशन इन द मेंडलिज्म, वेरिएशन इन द एक्सटेंशंस ऑफ द मेंडलियन जेनेटिक्स। मेंडल ने ये कहा था कि देयर आर टू फैक्टर्स, देयर आर टू टाइप्स, देयर आर टू फॉर्म्स ऑफ द फैक्टर्स व्हिच आर कंट्रोलिंग द ट्रेट्स। वन इज द डोमिनेंट एंड सेकंड वन इज द रिसेसिव। डोमिनेंट वो होता है which can exhibit itself in the dominant as well as the recessive form, which can dominant can exhibit its effect in the homozygous form as well as in the heterozygous form, while the recessive can only exhibit its effect in the homozygous form. Allelic variation, if we look on the dominants, dominants can be of several types. There could be a codominant, there could be an incomplete dominant. Alleles instead of only two alleles, there could be a more than two alleles in a population. According to the Mendel, an allele is a dominant if it has the same phenotype effect in the heterozygous as in the homozygous state. That is, in the genotypes capital A small a and the capital A are phenotypically indistinguishable. We cannot distinguish between the effects of capital A and small a and capital A capital A. You can say we cannot distinguish between the effects of homozygous and the heterozygous form of the dominant allele. Sometimes, however, a heterozygote has a phenotype different from that of either of its associated homozygote. Say, for example, flower color in the snapdragon is an example. On the basis of this variation, the dominants can be divided into the two forms, two major forms. One is the incomplete dominant, and second one is the codominance. In incomplete dominant, flower color in the snapdragon is the example. Heterozygote in this dominant is also known as the semi-dominant. The flower color in the snapdragon A majus is the example. Heterozygote has a phenotype different from that of the either of its associated homozygote. It depends on the amount of product made by the color gene. You can say in the incomplete dominance, this is also known as the partial dominance. The allele for red color, say for example, the flower color of the snapdragon, the allele for the red color, capital W, is therefore said to be a incompletely or partially dominant over the allele for the white color, say for example, small w. In the heterozygote, instead of showing in the red or the white state, the color of the color intensity is dropped down. Say, for example, it produces the pink color, which is in between the red and the white color. So we can say during the partial dominance or during the incomplete dominance or during the semi-dominance, the intensity of the product is dropped down. The intensity of the product is lowered down, or there maybe the different intensity of the product. When the heterozygote's phenotype is midway between the phenotype of the two homozygotes, as it is here in the partially dominant allele, is sometimes said to be the semi-dominant. The second one is the codominance. In the codominance, both the allele show their effect when they are in the heterozygous state. This is known as the codominance. The example of this dominance is the MN blood type of the human. MN blood type of the human. Uh, Human blood type MN, in this blood group, one serum called the anti-M recognizes only M antigen on the human blood cells. Another serum called anti-N recognizes only N antigen on these cells. These two alleles appear to contribute equally the phenotype of the heterozygote. The ability to produce the M and N antigen is determined by a gene with two alleles. Homozygotes for the M allele only produce the M phenotype. homozygote of the N allele can only produce the N phenotype, while the heterozygote Mn while the heterozygote Mn can produce the Mn phenotype. So M allele and N allele, when they come together, show their effect. It means both of the alleles can show their effect when they come together. Such type of dominance is known as the codominance. Moving toward the multiple alleles, the Mendelian concept that the gene exists in no more than two allelic states had to be modified when genes with three or four or more alleles were discovered. Dear students, remember when we talk about the multiple alleles does not mean that these multiple alleles are present in these multiple alleles are present in the a single organism. It means the presence of the multiple alleles in a population. So a population can have more than one allele for a single trait. Genes with the more than two allelic states are common. Say, for example, four alleles: C for albino, CH for Himalayan, CCH for chinchilla, C positive for the wild type, control code color in the rabbit in different ways and are mutant of the C gene. Wild type alleles such as C positive usually refer to the most frequent alleles in a population. The example of the multiple allele is the fur color of the rabbit, the coat color of the rabbit. There are four alleles: C albino, CH Himalayan, CCH chinchilla, and C positive, the wild type. White hairs over the entire body. If you look on the combination of these genes, if C plus, the coat color over the entire body, the colored hair over the entire body will be present. If CH, white hair with the black tips, and CH, black hair on the extremities, and CC is the completely white, completely white. Actually, it is completely white. In this example, C plus C plus is dominant over CH. CH is dominant over CH, and CH is dominant over C. So remember, I told you that multiple alleles mean that existence of more than one allele in a population. So, for example, the C plus, when crossed with CC, it will produce C plus. It will produce C, and when both of these will combine, they will make the heterozygous wild type. And when this small c is crossed with, say, for example, CH, CH. So this is the allele 1, this is allele 2, and this is allele 3. So it will give the gamete CH, and when it will combine with this one, it will give CH, C, Himalayan. Here you are looking, it is containing two alleles, it is containing two alleles, it is containing two alleles. After fusion of the gametes, the heterozygotes are also containing the two alleles. So it does not mean multiple alleles. Multiple alleles does not mean the presence of more than two alleles in a in a single individual. Being a 2n, a single individual can only have a two allele. The presence of the multiple allele, it means there may be more than two alleles in a single population. Second example of the multiple allele is the human blood type. The human blood type, the genes responsible for for producing the A and B antigen is denoted by the letter 1. Okay. Before going to the human blood type, There is another example of the multiple alleles, which is the uh, eye color of the Drosophila. There are more than two alleles for the eye color of the Drosophila. Uh, I'm here writing for all those colors. Say for example, the eye color for the completely white, and here the red, or you can say red, or you can say white coffee. Say for example, there are more than two alleles. There is actually a range of colors for the for the eyes of the drosophila. This is the mutant white. From mutant white, there is a second type, white apricot, then white eosine, then white, for example, cherry. After the white cherry, there is white coral. After the white coral, this is the white color. After the white color, there is the white wine. After the white wine, there is the white blood color. After this, there is the white carrot, and after the white color, the white coffee color. So if you look, there are 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10 alleles only for the eye color in the Drosophila. Here you can see that multiple alleles provide the range of phenotype. Here you can see provide the range of phenotype. There is the division of the range of the phenotype. You can say if after this one is the wild type. Say for example, wild type. Here is the wild type. So here is the wild type, and here is the complete mutant. Mutant. We can see from the mutant to the wild type, there is a range of phenotypes. So multiple alleles provide the range of the phenotypes. Okay. Now we are moving toward the blood group, human blood type ABO. ABO is an excellent example of the excellent example of the you can say multiple alleles. The gene responsible for producing the capital A and the B antigen is denoted by the letter I. There are three alleles IA, IB, and this I is small I. You can say IA allele specifies the production of A antigen. IB allele specifies the production of B antigen. I allele does not specify any antigen. So in the homozygous state, I cannot produce any antigen. So the blood type will be the O. The IA and IB are the codominant over each other, and IA and IB are dominant over the small I. So a small I is the recessive. Here you can see if A is homozygous or heterozygous with the I, it will produce the A blood type. If B is the homozygous or heterozygous with I, will produce the B blood group. IA and IB will produce the AB blood group, and II and O will produce the I in the homozygous state will produce the O blood group. So this is the one of the most firmly established series of the multiple alleles in the human involve the genetic locus controlling in the blood type. Actually, the ABO locus control the type of the glycolipid found on the surface of the erythrocytes, apparently by specifying the type of glycoside transferase. Syn glycoside transferases. The specific type of the glycolipid on the red cell surface, it provides the antigenic determinants that react with the specific antibodies present in the blood serum. Human, like all other mammals, produce antibodies and circulate them in the blood serum as a defense mechanism against the foreign substances. Fortunately, no antibodies are are synthesized in that react with the antigen present on the individual's own cells. So, for the own cells, then antibodies cannot be produced. However, when type A blood and type B blood are mixed, the anti-A antibodies in the type B blood serum react with the antigen on the type A blood cells type and vice versa, which produces agglutination or clumping of cells. Cross-matching blood type to determine compatibility is thus essential in blood transfusion. In this process, blood donors and recipients are tested for the presence of antigens and antibodies that are incompatible. The second example for the multiple alleles is the Rh factor. Regarding the Rh factor, there are three hypotheses. Regarding the Rh factor, there are three hypotheses that this factor is biallelic, this factor is multiallelic, and this factor is multigenic. Biallelic means there are only two alleles, capital R and the small r. Capital R, capital R is responsible for the creation of R positive, and small r is responsible for creating the Rh negative. It can so according to this hypothesis, the capital R is dominant over the small r in the heterozygous R positive will be expressed. While for the multi-allelic hypothesis, this hypothesis explains that there are around eight alleles. There are around eight alleles which are responsible for creating the Rh positive or the Rh negative form of the blood. Among these eight alleles, four alleles are capital R and four alleles are small r. Among the capital R, the four alleles are R1, R2, R0, and RZ. And among the small r, there are four alleles R, after the R there is second allele which is R prime, then R double prime, and to distinguish from the capital R, RY. So this is the second hypothesis that eight alleles are responsible, and these eight alleles can produce the various types of the Rh positivity. While the multigenic hypothesis explains that there are three genes which are responsible to create the positivity in the R. These alleles are the C, D and E. C, D, and E. Their various combinations can explain what type of the R what type of the R will be over there. What type of the R positive or R negative will be there. According to this hypothesis, this the Rh positivity or negativity is controlled by the three genes which are C, D, and E, which are closely located to each other on the same chromosome. Such closely associated gene would be expected to act like alleles in most of the situations. Um, and it was explained that Rh positivity, or we can say the capital R or small r, is dependent upon the dominancy of the D allele. It means that capital D, if in the combination the capital D is the present, the type of the blood group will be R. The type of the blood group will be the capital R. Say for example, C, D, and small e, small e, and small c, capital D, and small e. Similarly, small c, uh, small d, small e, and capital C, small d, capital E. Because here in both of these types, the capital D is present, this will produce the capital R type. Because here the small d is present, it will produce the small r. This is all about the multiple alleles, and I hope this is enough for today. We will join for the next class for we will join for next class tomorrow, inshallah. Good luck to all of you.